This video provides a clinical and forensic guide to taphonomy and postmortem changes. It outlines the early postmortem changes, including Algor, Livor, and Rigor Mortis, and the various stages of decomposition. Somatic death, or the collapse of the tripod of life, is the permanent cessation of vital functions. This collapse involves the failure of the brain, leading to unconsciousness and loss of reflexes, the heart, causing a stoppage of circulation, and the lungs, resulting in cessation of respiration. At the moment of somatic death, immediate changes occur. These include complete loss of circulation and respiration for over 4 to 5 minutes, leading to the loss of voluntary power, cessation of oxygenation, and halting of blood movement and heat transport. The golden window of cellular survival occurs 1 to 2 hours after somatic death. Tissues briefly survive anaerobically, retaining residual activity, such as ciliary movements. Muscles exhibit primary flaccidity, relaxing as ATP permits cross-bridge splitting. Contact flattening also begins as the body rests on a surface. Early surface and ocular manifestations occur within minutes to 2 hours. The skin becomes pale and loses elasticity. Intraocular tension plummets, causing the eyes to look sunken. Pupils initially dilate, then constrict with rigor mortis onset. Tache noire, yellow triangles on the sclera, may appear if eyelids remain open. Algor Mortis, or cooling, spans 1 to 20 hours, following a sigmoid curve of heat loss. Postmortem heat exchange occurs via conduction, convection, and radiation. Core temperature measurement is a primary metric for estimating time since death in the early postmortem window. In some cases, body temperature may remain raised for the first 2 hours due to postmortem caloricity. This can be caused by conditions like thermoregulatory failure, excessive muscle activity from convulsions, or hyperbacterial activity from severe fevers or septic conditions. Livor Mortis, or the staining of death, occurs from 1 to 12 hours. Circulation ceases, causing blood to stagnate and gravity to pull red blood cells into dependent capillaries. This results in bluish-purple or purplish-red discoloration. Areas compressed by weight remain pale. It is medico-legally important for indicating posture and estimating time since death. Lividity becomes well-developed within 4 hours. By 6 hours, blood coagulates in the capillaries, fixing the stain. Distinct colors can indicate toxicological factors, such as cherry red for carbon monoxide poisoning and pink for hypothermia. Rigor mortis, or stiffening, is a highly predictable physicochemical change occurring from 1 to 36 hours. After death, ATP is destroyed, leading to an inexhaustible actomyosin complex that causes muscles to shorten and harden. This change is used to bracket the time since death. Rigor mortis follows a proximo-distal progression known as Nistain's rule. It starts in involuntary muscles within 1 hour and progresses to the extremities. It disappears in the exact same order as tissue decomposition begins. Climatic variables significantly influence rigor mortis. In India, onset begins 1 to 2 hours after death, developing fully in 2 to 4 hours. Duration varies greatly between summer and winter. Heat accelerates the biochemical breakdown of ATP, while cold delays it. Cadaveric spasm is an instantaneous rigor occurring at the moment of death, unlike the 1 to 2 hour onset of rigor mortis. Cadaveric spasm is usually restricted to a single muscle group, indicating the exact mode of death, often associated with severe trauma. Stiffness is not always due to actomyosin complexes. Conditions simulating rigor mortis include heat stiffening, where muscle proteins coagulate above 65 degrees Celsius, and cold stiffening, where tissues freeze solid. Gas rigidity, from putrefactive gases in advanced decomposition, also forces limbs into stiff positions. The onset of decomposition involves late changes from days to months after death. This includes autolysis, or self-digestion by the body's own enzymes, and putrefaction, the breakdown of tissues by exogenous bacteria, which becomes grossly evident after rigor mortis passes. Putrefaction is largely driven by Clostridium welchii, an anaerobic bacterium from the intestines. This bacterium produces lecithinase, an enzyme that hydrolyzes cell membranes, causing massive postmortem hemolysis of blood. The first visual sign of putrefaction, visible in 12 to 48 hours, is a greenish discoloration over the right iliac fossa or cecum. This area is heavily loaded with bacteria and is closest to the surface, making it an ideal starting point for decomposition. Between days 2 and 10, decomposition progresses visually. Gaseous swelling occurs in 18 to 48 hours, leading to bloating and postmortem purge. Marbling, or superficial veins staining greenish-brown, develops in 36 to 48 hours. Skin slippage and blisters follow in 2 to 3 days, and colliquative liquefaction begins in 5 to 10 days. An environmental exception to putrefaction is adiposury, or saponification, which takes 3 weeks to 6 months. In warm, moist, anaerobic conditions, body fats are converted into insoluble soaps by intrinsic lipases and Clostridia enzymes. This yellowish-white, waxy substance preserves facial features and injuries. Another exception is mummification, occurring from 3 months to 2 years. In hot, dry air, rapid dehydration prevents putrefaction. The skin becomes shrunken, dry, and leathery, adhering tightly to the bones. This halts decay and permanently preserves anatomical structures. Forensic pathologists estimate time since death by synthesizing the clock, reading the intersection of all taphonomic changes. This includes body temperature, stiffness, and the presence of putrefaction signs like green discoloration, as shown in this graph of postmortem changes.